frozen.
Aw, man!
Hi, so welcome back to Science Savvy, and today we're going to come back with a new video. And today we're going to do not diploma, MYP, instead of IGCSEs. So as we know, as we go into grade 11, we have the MYP tests for and that's going to cover C1.1, 1.1, 1.2, and 1.3, right? So let's break it down. As I heard, this syllabus doesn't really give a base into sciences. So because of the lack of base, it tends to be a bit hard for people. So let's start. Our first topic is going to be enzymes, C.1.1.1. And I think this entire video is just going to be this one because it seems like the chapters are a bit lengthy. So let's start. Enzymes, maybe not the calligraphy but the handwriting. Okay, so let's start. The resource that I'm using to teach this will be linked down in the bio, so look there if you want to. Now we can start with enzymes. So what are enzymes, right? Because, well, as we go into the chapter, we need to know what the main term is, right? So what is an enzyme? Enzymes are biological catalysts. So this is the most brief description you can give of what an enzyme is. It's a biological catalyst. Over here it's written enzymes are biological catalysts that speed up the rate of chemical reactions in living things. So what I've understood is that because of the big terms, people tend to get confused with what they actually are. So before we move ahead of this first line, we have to see what a catalyst is, right? What is a biological catalyst? I mean, the biological makes sense, you know? It's part of biology, it's part of nature, okay? It's not man-made, it's natural. But what is a catalyst, right? In truth, catalyst is more of a chemistry term. So if you've taken chemistry, you might have already heard about it, but I'm not sure if it's like that in IB MYP, sorry. I don't know if it's like that in IB MYP. But in other syllabuses, we tend to cover what a catalyst is. For many grades, actually, starting grade 7, 6, we kind of have heard of this word already. So, what is a catalyst? A catalyst is a substance that speeds up the rate of reaction without itself getting used up or changed in the process. So what does this mean? That's like a lot of words, but like what is it supposed to tell us, right? So in this would be a substance that speeds up the rate of reaction without itself being used up or changed. So in that case, your substance or like action in this case would be your sibling leaving the room, right? So they haven't been changed, they haven't been used up or anything, right? They're just there and they're still fine, right? Another example is yeast. So when you're making bread, this is with actual. Another example would be yeast. So this is also with biological catalysts, which means they use enzymes to become fermented. So when the bread wants to become bread, right? Like the yeast is ready to change into bread, they tell the enzymes within them to start using carbon dioxide, and it's a whole lot of a process, but it's because of enzymes that they turn into bread this quickly. Otherwise it would take longer time. Even in chemistry, most of the experiments we do, we have catalysts, which is an example of which was the industrial process of ammonium, Haber process, also known as how we got the NPK, agricultural tree, etc. So it's just the same thing as before. now we can look into our next term, which is they are globular proteins. Okay, so we're talking all about enzymes. Okay, even now I know we had a little line about catalysts, but enzymes are biological catalysts. So first we defined it, now we're describing its shape. In the definitions written over here, it's also the not used up or changed process is also written up over there, but we can first look into the shape. So they are globular proteins. Now, first, let's break down the sentence because it starts with they are globular proteins, meaning they're polypeptide chains folded into a rounded three-dimensional shape. Break this down. That's a lot of information. When we start, we hear globular proteins. What is a globular protein? And what does it have to do with enzymes? First off, enzymes are proteins. So you wouldn't have enzymes without proteins, okay? Now, let's think of enzymes in the case of ourselves. If we look at our human digestive system, to break down lipids, which are also known as fats, you have lipase, which is an enzyme. Okay, let's talk about this from the IGCSE point of view, okay, because, well, that's what I study. Okay, what happens in my curriculum, what they say about it is that an enzyme, suppose it's like this, okay? This is the enzyme, and this is a molecule of lipids, okay? Now what the lipid does is that, this is lipase by the way, because lipase is an enzyme that breaks down lipids. Now what happens is that the lipid molecule goes here and it connects. So now it looks like this. It doesn't enlarge in, sorry for that. Now why does it look like that? Okay, what is happening over here? So the fact that this whole shape is made over here gives it a term. I mean, the shape itself has a term, which is the enzyme substrate complex. Okay, so what does that mean? Okay, so this is a substrate. A lipid is a substrate and lipase is an enzyme. Okay, so they both combine to form a complex. That's it. Pretty simple, right? Now, what happens next in this process? Well, this area right here where both of these meet is also called the active site. Okay, another very important point. Okay, now what is the active site? That's the place where the enzyme acts upon the lipid. Okay, so this is in between a whole sea of liquids. You know, our body has lots of liquids inside it, like bile, stomach acid, etc. Right? So what's happening over here is that the lipid is getting broken down by the enzyme. Okay, what the enzyme is doing is it's breaking it down, breaking the substrate down to gain more surface area for the enzyme to act upon it. And when it acts upon it that way, like, you know, if you have a higher surface area, you have a higher rate of reaction. Okay, that's another thing in chemistry that we've learned. Okay, so if you think about it, you have a solid shape, okay, and you have powder. Won't the powder react quicker than a solid shape? Like, if you think about it, again, another example, sugar cubes versus sugar powder. Wouldn't the powder melt quicker in tea in comparison to sugar cubes, right? When you look at from that aspect, you can understand what and how well an enzyme actually works, right? Now, enzymes cannot be made without ingesting proteins, okay? So that's why lots of people say, okay, eat proteins if you want to grow up, if you want to grow strong, grow this way, grow that way, eat proteins. Because they make up your enzymes. Because enzymes are proteins, you must eat more proteins so that you can have more enzymes. And when you have more enzymes, you have more ability to like digest your food, have faster reactions, grow faster. See? It's all connected, okay? Now, when you look at globular proteins, you know that enzymes are proteins. That's why there's proteins written over there in full bold. And what are globular proteins? It's a globular proteins. Over here, there's more descriptions on globular proteins. It's like their polypeptide chain folds into a rounded three-dimensional shape. What is that? So a polypeptide is a chain of amino acids. So what's an amino acid? proteins, when they're broken down, they make amino acids. If you look at that sense, polypeptide chains are just a different version of proteins. Polypeptide chains fold into rounded three-dimensional shapes. When we look into what are globular proteins, we can look into their structure. The main base structure of a globular protein starts off as small circles, and then they start turning into coily shapes, and then they become a whole mixture. Quite literally look like this. They're one of the two types of proteins, and we'll look into that a lot later, but this is how they start off the chapter with. Now, let's look at the next one, and that is activation energy. That is also mostly a chemistry term, which is like, of course, overlapping with biology right now. And when we look into this, activation energy is the least amount of energy you need to start a chemical reaction. So if you just try to light a fire, it won't just immediately spark up, right? You have to make sure you get a certain amount of friction between two sticks to make sure that fire lights up. Or if you're trying to start a car, you have to make sure it reaches a certain degree before it actually starts up, right? So that's basically what activation energy is. The minimum amount of energy required to start a chemical reaction. Now let's take a look at the next term. How catalysts work. Lowering the activation energy. As we've said, activation energy, minimum amount of energy required to start a reaction. So every reaction needs a starting push of energy, right? And that's called the activation energy. And enzymes lower this, which is why the reaction happens far more easily and far more faster. This is also why reactions can work at like 32 to 34 degrees Celsius within our bodies, from around 37 degrees Celsius, correction. Another thing we can look into this is the diagram we see in this page. I've added over here. This is a really important diagram actually, and it showcases how the enzyme's activities working with the activation energy. So this green line is supposed to be the normal reaction, while the blue line is with a catalyst, with an enzyme. Let's see the difference. Showcase this difference a bit more easily, because we're like, okay, where are we seeing the difference, right? So if we make a red line over here, which was the starting point, right? It started over there, so let's make a big line over there. This way upwards, when we look at how high the line has reached, that is how much activation energy was used up. So over here we can see the line has reached till here. We can see the visible difference in how much activation energy was used up, right? Activation energy was required, okay. And over here actually it's the products of the reaction. Okay. So by this spot, the reactions have ended, and we're looking at the products that we've gotten from this particular reaction. I hope you understand this, okay? You don't ask me questions in the comments. We're gonna move on to the next, which is enzyme specificity. This is related to the enzyme substrate we had talked about before. Let's just redraw that 1 second. Here we've redrawn the enzyme substrate complex. What we're talking about here is, we already have a gist of what this is. How does it tie to enzyme specificness? Well, what happens here is that this enzyme, suppose the enzyme is, we can take an example of lipase, and the substrate is lipids. We can also take another example enzyme, suppose that is carbohydrates. Now carbohydrates, as the name suggests, it's for breaking down carbohydrates. Now when you look into this entire thing, why did I draw two enzymes, right? What's she gonna tell us, right? What's she gonna tell us? The enzyme lipase and the enzyme lipids are complementary to each other. Okay, so if another enzyme tries to come in, suppose if lipase tries to, lipids try to go into a carbohydrate, that's not gonna work, right? Because they're not the same sheet, they're not complementary to each other, right? Complementary as in if this is a jagged line, this is another jagged line. So they're complementary to each other. They're meant to fit together like a puzzle piece, right? So if a puzzle piece is connected with a puzzle from a different puzzle piece set, that's not going to work, right? The entire concept here is just a bunch of puzzle pieces. Right? A certain enzyme cannot pair with any enzyme other than the enzyme that it's made for. So lipase can only pair with lipids. Lipase cannot break down carbohydrates or vitamins or any other type of compound or substrate. Why are enzymes important though? Enzymes, as we've said before, help speed up reactions. If we didn't have enzymes, all the reactions in our body would be so slow. Now for C1.1.3, they flash you with really, really big terms in the start itself. So of course it can be a bit confusing. Now we split metabolic reactions into two types. We've discussed what metabolism is in my previous video for IGCSEs, but let's talk about it over here. So what is metabolism? Metabolism is all the reactions that happen in our body. And over here we split it into two types. So that is anabolic reactions and catabolic reactions. Okay. Now what are these? Anabolic reactions. Now anabolic and catabolic reactions are two different spectrums. So they're both opposites of each other. Now what happens with anabolic reactions is they build larger molecules from smaller molecules. They build larger molecules from smaller molecules. So what does that mean? Suppose when we look at proteins, as we said before, amino acids. So if we have a few amino acids, so when we look into amino acids, we make proteins by making chains of these amino acids. So suppose it looks like this. These are the amino acids. Now to make it proteins, we turn them into chains. So making smaller molecules like amino acids into larger molecules like proteins is what an anabolic reaction is. Okay? And it requires really little amounts of energy. Okay, so if you let's just make a supposed value over here. This requires 5 joules. Okay? Now, in catabolic reactions, they break larger molecules into smaller ones and release energy. Okay? And the thing with these is that it requires an input of energy. So without giving energy inside, nothing's happening. That's why in certain metabolic processes, if you think about it, we have to eat food, right? Food serves as energy for these metabolic programs and processes in our body. And when we look at catabolic reactions, on the other hand, they break down larger molecules into smaller ones and release energy. So, for example, if you take amino acids and proteins again, so when we look into catabolic reactions and the amino acids proteins example, we see that from proteins to amino acids is what happens when there is a catabolic reaction. We see that during a catabolic reaction, they break down the larger molecule into a smaller one, and then in that process they release energy. So it's much like this is a larger molecule, of course, and then these little chains, these lines in between, they're broken down and then made into smaller pieces. So it's basically a breakdown or a make kind of reaction, you know? So like it's if you're making Legos, right? You're gonna click it on, which makes a little bit of a sound. You need to. So suppose you're using Legos. When you're making Legos, you have to click both the pieces on, which of course needs energy to do. And when you're removing glucose, of course there's a force that's pulled outwards, right? So it's releasing energy, right? So it's release energy requires energy input. Catabolic means you break down big to small. Anabolic means small to big. Okay? Now this can be kind of confusing, so I recommend using a mnemonic. For example, now anabolic reactions build complex molecules from simpler ones using energy supplied by ATP. So what is the ATP? ATP is what you get when your body breaks down food. It's basically energy. Energy from food is basically called ATP, okay? ATP's full form, on the other hand, is adenosine triphosphate, but I don't think you guys have to remember that. Now, anabolic reactions are usually condensation reactions, which means they release a water molecule each time two subunits are joined, okay? So this example of a condensation reaction, which again from chemistry would be, so an example of condensation reactions would be in esterification, which is forming an ester in chemistry once more. So in chemistry what happens is something called a carboxylic acid and a alcohol, they mix together to give out an ester and water. So when a reaction such as acid plus alcohol makes a product and water, it's called a condensation reaction. Okay, pretty easy, very straightforward, just gotta remember. Okay, now the next thing that they talk about over here is that photosynthesis. The next thing they talk about over here is protein synthesis. So what's protein synthesis? So protein synthesis is basically amino acids. Okay, so amino acids into polypeptides, which are also known as proteins. Okay, this right here is that reaction, just that they've removed this name and made it polypeptides. Okay, both are the same thing, just a different form. Okay, and this process is called protein synthesis. And to do this is a condensation reaction. Each peptide, polypeptides, so each peptide bond is made using the condensation reaction. Okay, next up we have the glycogen formation, glucose into a storage polymer. So glycogen is a storage molecule. It's a large molecule in our body that's formed using glucose and stored. So when we exercise, where do we get energy from? We already ate food, but where do we get the rest of the energy that we're using when we're exercising from? That's from glycogen. So glycogen, what happens with glycogen is that glucose is taken in using food, yes? Now that glucose in the food is then converted into glycogen and then stored in the liver. Okay, so this is a small molecule, this is a large molecule. Okay, or complex and simple is another way to describe it. Okay, so glucose is a simple molecule, glycogen is a complex molecule, which means it's larger. Okay, and then you store the glycogen inside the liver. Okay, now when we're exercising, what happens is the brain contacts the liver and tells them to break down this glycogen once more, and then the glucose is distributed through our body via blood vessels. Okay, so even when we look up above over here in ATP, right? So ATP is actually transported as well through our blood vessels. So most of these nutrients, anything that we have to transport across our body, water, nutrients, energy, etc., all through the blood. Okay, it's a transport system. Now we got glycogen formation as well, photosynthesis, which is in plants. Okay, so let's just write this down. So photosynthesis is an anabolic reaction. because it builds large glucose molecules from small carbon dioxide and water molecules. Okay, it can also be a condensation. It's not a condensation reaction, but it is an anabolic reaction. Okay, because when you look at photosynthesis, you get 6 CO2 plus 6 H2O, which is equal to C6H12O6 plus O2, right? So in this, you're not getting water as a product, right? It's in the reactant side. So this is the reactants as the products, right? There's a clear marking differentiation. And water is one of the reactants. It's not the product. So it's not a condensation reaction. So you could say that not all anabolic reactions are condensation reactions, but most condensation reactions are anabolic reactions. Now, for photosynthesis, rather than ATP is like the energy source in humans, but for photosynthesis, plants of course, their energy source is light. Next, we see catabolism. Let's explain this side of this entire big table. Catabolism, breaking complex molecules into simpler, smaller ones and releasing energy. Into simple, smaller ones, and release of energy happens through this process as well. So they're usually used by hydrolysis. Okay, they usually work by hydrolysis, which is the exact opposite of this side. So when you look at condensation reactions, what happens? You have a product of water. So water is given away, right? So it's taken out of what you're getting, right? Meanwhile, over here, what happens in catabolic reactions is that they add water to break the bond. Now, an example of this given over here is peptide bonds into a polypeptide into amino acids. Okay, so that process is called this is protein synthesis, this is protein hydrolysis. Okay, so in mnemonic you could use to memorize these two that I forgot to tell you guys before is that anabolic adds, catabolic cuts. Okay, so when you're looking at a catabolic reaction, they cut out stuff. If you look at an anabolic reaction, they add stuff into one place, you know? So that kind of makes sense. Now we go back to hydrolysis. Over here we can see protein hydrolysis. So the next thing we can look at after polypeptide to amino acid, which is called protein hydrolysis, which over here I add a few to break down, is digestion. So digestion, what happens in digestion is that we hydrolyze food into a what happens in digestion is that we hydrolyze food into absorbable units. Okay, so during digestion, proteins, carbohydrates, lipids, they're all taken, added. During digestion, what we do is that our bodies take in all the proteins, carbohydrates, lipids, and they add water to break down into monomers. Okay, so monomers is the simpler version of the complex molecule. Okay, so amino acids is a monomer. Okay, so for example, protease breaks down proteins into amino acids, and amylase breaks down starch into maltose. Okay, so it's quite simple, but if you have any questions, please mention those in the comment section and I will reply to you guys. Okay, now cellular respiration, a catabolic process. What is cellular respiration? It's when we breathe in and use oxygen to break down glucose into smaller molecules. Okay, the energy released is called ATP, of course. As we were saying, cellular respiration, in this we can see that there's going to be C6H12O6 plus oxygen, right? And that gets transformed into 6CO2 plus H2O plus ATP, right? So in that we can see that the glucose, C6H12O6, is broken down using the oxygen, 6O2 given out there, and it makes ATP and other stuff. So, other products. So, look further into this. There's always supposed to be it's important to have a balance between catabolic reactions in the body and anabolic reactions in the body. And that's because if there's no balance, it doesn't ease out how fast we grow or how fast we repair. Because anabolism builds proteins and other molecules needed for the tissues, and energy for doing that is supplied by catabolism and homeostasis. Now there's a requirement for balance if we want our body to continue being our body is a complex. Now there's a requirement for balance between these two topics, anabolic reactions and catabolic reactions, because if our body did not have a balance between these two, we would malfunction. Let's take this example: growth and repair. Growth and repair are done by anabolic reactions. They have to build proteins and other molecules needed for new tissue. But look at anabolic reactions. For an anabolic reaction, we need ATP, which is energy. Okay, so for that, catabolic reactions supply ATP using cellular respiration. So they're both interconnected. Without the other cannot function. Okay. Another thing is that there is something called homeostasis. Okay. Now homeostasis is what balances the two to keep internal conditions stable. Without homeostasis, we would not be stable. We would, like I said before, malfunction. And if internal conditions aren't kept stable, our body's gonna get sick. We're gonna have autoimmune diseases, etc. In the structure of an enzyme, there's one more thing that I have not mentioned, and that's how there are small amino acid side chains that connect the substrate to the protein to the said substrate. So these are amino acid side chain. Pretty simple, pretty easy. You just have to make sure you remember this stuff. By the way, we have reached about 1.1.5. Yeah, so far it's very little content, just that certain websites like to make it look really, really difficult to climb through. Okay, now binding is not a rigid lock and key event. It's the interaction between substrate and active site to allow induced-fit binding. So what this means is that induced-fit binding, okay, that's a big term. What is that now? Okay, let's just make a box over here so we can add more information. So induced-fit binding. I have a website that also talks about certain questions you'll get from each and every kind of topic in this, and including subtopics, by the way. So I'll leave the link for that in the bio. Substances in the reaction are changing shape to make sure they fit inside. Like when you're taking two pieces of rubber, okay, and you tear it apart and then you're trying to keep it back in. Okay, so first you have to shake it a bit, make sure it fits, or it's like when you're making a puzzle and then adding it into the frame. So you have to make sure it fits, wiggle some pieces in, move some stuff around. So basically that's what happens here. Okay, so they're making sure the binding is happening by changing the position and shape a little bit, okay, to improve catalysis. So catalysis is referring to catalyst, just the process version, Coming back to catalyst, a substance that reduces the rate of reaction while itself not being changed or used up in the process. So when a catalyst is acting upon the substrate, catalysis, I don't know. But yeah, so in this website we have a diagram, and in that we get, first we see that the substrate is approaching. Okay, so this is the enzyme, and the substrate decides to approach. Okay. Second, we see that there's weak interactions that form up. So when we look here, this is the piece again. Weak interactions. They could break any moment now, you know. Then third step in the whole cycle is how the products are released. Third step in the entire process is how the products are released. So we can see that now with the products have released by this little shape that they've given in. Okay, and in the third step, induced fit stresses bonds. So this little symbol over here shows that they're fit together now. Okay, in the actual diagram they're much closer, but we just wing it for now. Next, fourth step is when the products are released. So we look at this and we see two break-in pieces and outwards. So the products are released now, and the process is almost at its last. And last but not least, enzyme restored. So now it's ready to repeat the process and do it once more. How does an induced fit help catalysis? Catalysis. These shape changes help the catalysis by stressing bonds. So it stresses the substrate till it breaks down, and then the enzyme goes back to normal because the substrate no longer fits within the enzyme. Now, here we see a role of molecular motion and substrate active site collision in enzyme catalysts. So, there's something called molecular motion, which is a random movement of molecules caused by their kinetic energy. And it's inside a fluid cell environment. Substrates and enzymes move, rotate, and bump into each other as molecules constantly jostle around. What does that mean? So I've written it down over here. Molecular motion. Random movement of particles caused by kinetic movement. We all have heard of random motion, right? So particles are always moving, and they're moving in random directions. Look at gas. When you look into chemistry and you've learned the movement of gas, right? So it's always in random motion. It's always bumping into each other. Random motion, random speed, etc. Random motion, high speed, etc. Okay, so that happens in fluids as well. And over here, have you seen any bacteria, like a close-up of bacteriums or anything like that? If you see that, you can see they're always moving in different directions, and when they bump into each other, they build up kinetic energy, which is then used to break down this. And over here, a substrate active site collision is an encounter where a substrate molecule contacts an enzyme's active site. Many collisions go nowhere. For binding to occur, the substrate has to get close enough and line up in a suitable orientation, which is why collision theory helps explain enzyme activity. So what's collision theory? Collision theory states that for a chemical reaction to occur, reactant particles must collide with sufficient energy to break existing bonds and correct orientation to form new bonds. So this is another reason why not all the molecules we eat are broken down. Some are just lost without being broken down because collision theory, of course. So we can actually manipulate how the theory of collision works, because if there is a higher substrate concentration, it makes sure there's more substrates, there's so many things bumping into each other, so it reduces the rate of active sites being formed. The rate of collision with active sites. So it overall reduces the rate of reaction. The same way, raising the temperature up to a point where denaturation could be occurring if you go any higher, increases molecular motion and therefore collision frequency. So what does this mean? So to increase the rate of reaction, what you can do is you can increase temperatures, because the hotter it gets, the more likely, the more energy supplied for the particles to move, right? There is more energy for the particles to have a higher frequency of collisions, and because of that, the rate of reaction increases. So what they said to the point where denaturation becomes important is because after a certain temperature, your enzyme will denature, which means it will change shape and it will just die off. Okay, so we don't want it to denature, right? So we need to make sure we make the temperature a set time, which is another reason why homeostasis, as we mentioned, and these reactions, we have to make sure our internal temperature is stable, right? To make sure our enzymes don't die off, they don't denature, etc. The next thing we will look at is specificity. So it depends on the active site structure. So if the active site is not made for the substrate, they're not going to collide or become one, right? Also, this is more of a 2D view of this. There are 3D views on the internet. You can look them up, or I'll probably post a photo around here. Denaturation disrupts catalysis. Okay, so let's take a break from these notes because they're kind of making everything jumbled in my opinion. So first off, we found out a higher rate of reaction can be caused by increase of temperature. Now, another way they can do that is using an increase of pH. Let's phrase this better, okay? This is a bit too confusing. Let's phrase it better. Now, there's two factors affecting the rate of reaction. So two factors that should be kept stable. Let's make it three, considering the concentration factor. So 3 factors affecting rate of reaction. This includes 1, it must be an optimum temperature. What's optimum temperature? So it's the best temperature for our enzymes to act upon. When you look at the activity of an enzyme, actually we can just go back here. Suppose we ignore this first time, we make it pH. This is a what if scenario? Time is not there, it's pH now. Not pH, let's start with temperature. And this is about 60 degrees Celsius. Now you see the maximum amount of, let's get rid of energy. So when we look at this kind of graph, usually they'll be looking like this. And then it goes down, okay? So what happens over here? Suppose this right here is about 32 degrees Celsius. So what's happening over here is that at 32 degrees Celsius is the highest rate of reaction, which means that is the optimum temperature for that particular enzyme to work upon, okay? And any time after that, you can see it's going downward. Suppose we can look into a graph, okay? So this would be temperature, and this would be rate of reaction, okay? Now, how it would look is much like this. Okay, so what this is how most rate of reaction and temperature graphs look like. So we can infer the optimum temperature from this. What is optimum temperature? It's the temperature in which the enzyme acts best at. Okay, so even when you look into this graph, you can see it's the highest rate of reaction is received at this particular temperature. Suppose it's 32 degrees Celsius, which is why our body tries to maintain a particular temperature and make sure that we're always at that temperature. And like our body uses shivering or sweating to make sure we stay in that temperature. Otherwise, the enzymes, as you can see, it's going back down, they'll denature. Okay, So basically, denaturing means they're, of course, dead. So the optimum temperature is the temperature at which the enzyme works best at. Now, just like this, there is an optimum pH. Okay, same kind of graph, just that the number might be 7, 8, 10, etc. So our body makes sure that these enzymes have the certain number of pH that they have using stomach acid or bile. So bile is a basic solution while stomach acid is an acidic solution. So using those, they manipulate the pH of the certain substance that they're going to deal with or break down. Of course, concentration rate of reaction is related to collision theory, because more concentration, more rate of reaction. Less rate of reaction, sorry. Because more concentration, less rate of reaction. Now, let's look at the notes once more. Interpreting the enzyme graphs as models. So these explain or predict the behavior of an enzyme. And I've shown you the most frequent shape. Of course, it could be much more pointy, for example, could be like this, etc. It depends on the enzyme. Now, what we look into next is 1.1.9 actually. Now we look at variables in enzyme experiments. So this is actually uniform. It's there in any kind of experiment you do. In IGCSE papers actually, we have an entire paper dedicated to experiments, and in that, this is one of the topics we look into very frequently. So it's dependent and independent variables. I'm hoping you guys have heard of it. If you haven't, it's alright. So this can be really confusing because you have to not interchange them, make sure you write them the right way. This way, that way, could be, you could accidentally get it wrong because you mix them up. Okay, so there is independent and dependent variables. I'll break it down how I made these easier for myself to do, and some things you have to note about these. I'm not sure if you guys will have to do it, but it's something we had to note down. Now, dependent variables is the outcome you measure. So dependent, it's dependent on the independent one. Okay, that's what I used because you measure what is dependent, right? So this is right now really confusing, I know. The outcome you measure, and independent variables is the factor you change. Now, how do we make this simple? It's so confusing right now. Okay, let's take that table, that graph we made before. So over here, let's make it pH, and it's rate of reaction, right? Now, in this graph, if we want to test for that graph, what do? We'll have to grab some samples of an enzyme and a substrate, right? And we'd have to add them together in a bunch of tubes, test tubes. Now, you would have to change the pH to make sure you can test the rate of reaction, okay? So suppose you add pH 7 over here, pH 8 over here, and pH 9 over here, okay? 3 test tubes, 3 different pHs, okay? And you add a indicator. Suppose if you were testing for carbohydrates, you could do iodine. So suppose you add iodine test into each of them, okay? Now iodine turns blue in presence of carbohydrates, okay? And it will be brown in presence of nothing, okay? So first off, what do? We change the pH in each of them while keeping the things you keep. We change the pH in each of them, and then we get a certain answer. Suppose this one worked 80%. This one worked. Suppose this one worked 80%, this one worked 0%, and this was 100% rate of reactions, okay? So it's how much was broken down we're looking here, okay? Now, what's the independent and dependent variable in this experiment? The independent variable is the factor you change. So what did we change here? We changed the pH. Okay? Now, dependent variable is the outcome you measure. So what do we measure here? The rate of reaction. So it's pretty simple if you practice. Okay? So you might want to practice this a bit. Just search up independent, dependent questions, etc. Actually, wait, no. Just Google up independent, dependent variable questions and it'll be easy. Another thing you need to keep in mind is control variable. Okay, so now control variable is a bit easier, I would say, in comparison to the other two. But of course, you have to make sure you can think of them. Because control variable is a factor that you keep constant. Okay, a factor kept constant. Okay, so it remains the same throughout the experiment and will never change. Okay, so suppose in the above experiment we did a example of the constant variable would be how we never changed the enzyme or the substrate or iodine, or we should keep constant the temperature because otherwise it would confound or like interact with the experiment and make our results invalid. Alright, so it's pretty simple, pretty easy. Another thing we need to know is calculating the reaction rate. So the rate of reaction is calculated over here by reaction rate N by T. So what is the N and T? N is the amount, change in amount of substance. So this is equal to change in amount of substance by the time interval, also denoted by this and this. Alright, so it's change in amount of substance by change in time. Alright, or amount of time. Now if you're measuring gas, it would be rate of reaction in gas. Let's write that down so we remember. It would be change in volume, gas volume, which is also measured in, you should remember the units, by the way. If you do not remember the units, you're not getting marks in maths, bro. This is the change in gas volume by the change in time. Okay, time is measured in seconds here, and the change in amount of substance is measured in mole. Okay, not molecules, mole. Okay. Now, in school enzyme practicals, you might see centimeter cube, second, like you can see this, maybe GS 1 minus, the minus 3 by the power of minus 3, S minus 1 by the power of minus 1, etc. depending on your measurement. Okay, so it depends on what your school will be saying. Okay, let's do it in centimeter cube, that cube, mole, cube, etc. Okay, it's just basic measurements. Now, practical examples of measurement. Over here we have a experiment given out for, it's 2, I feel like I've said this before, but if I haven't, just see it. Okay, so 2H2O2 is equal to 2H2O plus O2. So this is hydrogen peroxide and it creates 2 water and 2 oxygen. So this example can show you what the dependent variable is or the independent variable is. So over here you can see the diagram that it gives off, and the diagram has it's basically a test tube, actually a conical flask. No, it's a conical flask with hydrogen peroxide into it and a catalysis source. So enzymes reduce the rate of the activation energy required. So an example of that would be if there is energy written here and there's progress. So it could start here, go down, and it would start here and go down. No way. Start here and go down. Okay, so this is without an enzyme, and this is with an enzyme. So there would always be a notable difference of how much lower the activation energy has become once an enzyme is added. Okay. Now let's look at our next topic. Where the enzyme works matter. Okay, so an intracellular enzyme catalyzes reactions inside a cell. Okay, so glycolysis counts as an intracellular pathway. Okay, this is kind of confusing, right? So intracellular and extracellular, might have told you guys before, it's when there's a reaction inside the cell and like inside cells, enzymes, etc. Inside cells, okay, cellular bodies more. And extracellular is when it's outside of the cell. So it's secreted by a cell and catalyzes reactions outside the cell. So an example would be chemical digestion in the gut, right? It works outwards, right? It's not working inside the cell itself. Now, the Krebs cycle is intracellular, glycolysis is intracellular, etc. So these are kind of tough topics and words right now, but it'll die down. It will probably be explained later on in different chapters, okay? So let's write that down. And another thing is that extracellular digestion is mostly... Another thing is that extracellular digestion is really important in when there's macromolecules, because they can't enter the cell membrane themselves. So to put the enzyme outwards and break them down before they can come back in would before they can come in would assist the body in being able to break down even bigger foods, because not everything's the size of a cell, you know. Even if you chew it enough, it's not going to turn into the size of a cell. Now, metabolic energy transfers are not perfectly efficient. Now, heat is energy transferred from warmer bodies or a region of cooler ones because of a temperature difference. Now, in metabolism, cells can't avoid producing heat because energy transfers in chemical reactions are not 100% efficient. So as the cells move, they use energy such as ATP or move along concentration gradients, etc. And this energy can transfer into heat. Okay, now this is how energy transfers work, you know. Some is lost off as heat, some is lost off this, that, etc. It's there everywhere you see. Like if there's a light bulb, it's not only producing light, it's giving off some of its energy as heat as well. So basically that's what our enzymes do too. Now this heat given off sometimes helps animals, mammals, birds, and other animals to produce metabolic heat and keep themselves warm in a constant body temperature. Okay, if heat drops too low, it can raise metabolic activity, and that causes shivering. So shivering, muscle contraction uses ATP, and using that, they increase the rate of metabolic activity, hence making you use more energy and generate heat. Some tissues make heat as well. So this includes when, such as in brown adipose tissue, which contains mitochondria and can oxidize substrates in a way that releases more energy as heat rather than conserving it as an ATP. So metabolism and temperature regulation are linked. In IGCSE, we learned this as homeostasis. It was a year 11 topic. So yeah. Linear pathways have a start and an end. So metabolic pathways, yes, they are linear pathways. Now these have a start and an end, of course. They can't. So glycolysis is an example of a linear pathway. It's when glucose is converted through a sequence of intermediates into pyruvate. A specific enzyme catalyzes in each step, so different enzymes altogether, and the molecules gradually changes rather than in one chemical loop. Now, pyruvate is used in our body pyruvate is used in our body to generate a net yield of usable cellular energy. So basically it helps make ATP. Okay. Now that was a bit confusing, I know. Even for me, actually that was a bit confusing because I haven't learned glycolysis yet, but it makes sense, you know? So to make ATP, they make glucose, a six-sided thing, into pyruvate, which has only 3 sides. So it's broken down into a smaller molecule in order to make ATP. Makes sense, right? Now, another thing is cyclical pathways, which regenerate an intermediate. Okay, this is a lot of words. Now, pathways. They're a bit confusing, so stay with me right now. Okay, so there is one kind of pathway, also known as linear pathways. Okay, they're the more easier one. Okay, so they follow glycolysis. Okay, an example of this would be glycolysis. Now we're gonna do pathways, and they're a bit confusing, so bear with me right now. Okay, so first kind of pathway we're looking at is linear pathways. Okay, so they're pretty straightforward. They have a start and they have an end. Okay, they start with one thing and they end with another. Okay, so an example of this would be glycolysis, and we've gotten a whole chain of what happens in glycolysis here as well. Okay, so it starts with glucose, and then an enzyme's there, it becomes a more subpart, and then again another enzyme breaks down more, enzyme breaks down, and then it finally becomes pyruvate. I don't know how to say it, but you know how to say it, right? So what happens here is that a 6-carbon molecule, a carbon sugar, okay, such as glucose, breaks down into 2 molecules of pyruvate. Okay, so this is a 3-carbon sugar, hence why 2 molecules, obviously. And it's pretty straightforward. To get this, we break it down. Okay, so pyruvate is transported in molecules and converted into something else, basically citric acid, and this initiates the Krebs cycle. Actually, another way it's used is in the absence of oxygen, it's converted into lactic acid. Okay, so this is not really meant for your syllabus so far, which is why it's not mentioned here, but it's good to know, you know, so in the future when you're like, oh yeah, what's that thing again, oh yeah, that's how it's used. It's always good to know the use of something, because when you know it, it's easier to connect everything together. The next type of pathway is cyclical pathways. Now these follow a cycle. Okay, suppose Krebs cycle. Now what happens in Krebs cycle? Okay, so it's a series of chemical reactions that the cells use to release stored energy from food. It's like the breakdown of food or something. Okay, so first they take the pyruvate, I don't know how to say it again, and they combine it with a 4-carbon molecule, okay, not carbon sugar, carbon molecule, okay, and then they form a 6-carbon molecule called citric acid, okay? And after that, it goes through oxidation. Carbon atoms are removed, released as carbon dioxide, and then there's a bunch of energy extraction, lots of mess, but we know the gist, right? And after it finishes, it repeats. So it's a cyclical pathway because it's never ending. It doesn't have a start or a stop. It's always running, no end, no stop, nothing. Okay, over here in the photo they've given, it's regenerated intermediate. Okay, there's CO2 coming out, reduced carriers, ADP also going out, carbon compounds entering in. So it's just a big cycle, never ending. Another thing we need to know is allosteric site. Okay, now what's an allosteric site? Allosteric site. So the last two things we need to look into in this topic are two types of inhibition, and we have a few examples of it too. We begin with feedback inhibition. Okay, so what is feedback inhibition? Okay, first off, what's inhibition? Inhibition refers to the process of slowing down or stopping a process. That makes it much easier, of course, even for myself. Now, when you look at inhibition, you're not like, that's a tough word. You're like, it's just stopping or slowing down something. First type, feedback inhibition. It's when the final product of a pathway binds to an earlier enzyme in the same pathway. It's kind of related to the linear pathways. So, it's suppose this product binds with this enzyme, that would be feedback inhibition. And this enzyme binds with an earlier enzyme in the same pathway to temporarily deactivate it. So what happens is that it binds to the allosteric site, Okay, so it basically acts as a non-competitive inhibitor, just that it's from the same pathway. It only works in linear pathways, of course, because it's the end product that comes to an enzyme in a earlier process, you know. And these can actually work in any type of pathway. It's kind of a complex thing, branched and complex metabolic networks, not just linear pathways, okay? Works in a lot of stuffs, but the basic idea is that the final product binds with an enzyme before it to make sure that the enzyme stops working and to prevent the production of more of the final product. Easy peasy and lemon squeezy. Basically, works as a non-competitive inhibitor, and it combines with the allosteric site. Okay, that's why it's a non-competitive inhibitor. Non-competitive inhibitor, allosteric site. If it's a competitive inhibitor, it's the active site. Two types of sites, don't forget. Irreversible inhibitors, on the other hand, they are when a inhibitor binds to an enzyme so strongly by using the usual methods, you know, usually when there's a competitive inhibitor trying to just bind in, okay? But they do it so strongly that it permanently disables the enzyme. So what happens with the transpeptidase and penicillin? Transpeptidase is the enzyme, by the way. So this is transpeptidase, and the enzyme, the substrate, penicillin, okay? So what happens is when the transpeptidase is a susceptible one, there's a irreversible covalent bond, okay? Irreversible covalent bond. Active site is chemically changed, by the way, and this stops cross-linking and causes a weakened cell wall. So when there's a weakened cell wall, there's lysis happening. Lysis is when a substance goes into the cell, weakened cell wall, and this causes the enzyme to stop working completely. Meanwhile, if it was a resistant bacterium, the active site would be there, penicillin would bind poorly, cross-linking continues, cell wall remains intact. So that's why penicillin works how it does. So the resistance through changed peptidase. Penicillin resistance can arise when bacteria have changed transpeptidase with altered active sites. So it's a structure-function relationship. A small change in enzyme structure can change inhibitor binding, and that can change survival of the said enzyme. Okay. Meanwhile, we're going back to feedback inhibition. There's an example for that too. So, in feedback inhibition, another example given for feedback inhibition is negative feedback. When the end product concentration rises, inhibition increases and production drops. Negative feedback is what helps make sure our internal temperature stays at 1 point, by the way. Now, over here we see isoleucine as the isoleucine production as the example. So over here with the photos they've given us, we see that threonine, which is a substrate, combines with threonine deaminase, which then of course goes through its process, produces isoleucine, and then isoleucine goes back to the threonine deaminase enzyme. It's like as if threonine joined with this enzyme, okay, and then it changed, of course. So it's like when threonine combines with this enzyme, and then it goes through the process, and at the end, this we can change it to isoleucine, and the isoleucine finds this one's allostric site and binds back, therefore stopping the production of isoleucine. And that is how feedback inhibition works. Pretty simple, pretty easy, just that these terms and bacterium names of all of this are going to be a bit confusing. I will be attaching my notes down in the link below. Be sure to check out Instagram too. We're probably gonna post some definition videos inside of the IB curriculum. First, we started off with IGCSE. If you're an IGCSE student, please check that out too. Thank you for watching this video. Hope it's entertained you guys. See you next time.